Right, so what we're doing is we're looking at CORMS, and CORMS is the acronym. The other one I've seen is something called Doris. But anyway, CORMS is the one that I'm used to. It's for when you're doing experiments, when you're planning experiments. Right, remember we said, let's just go back. There's three types of variable. Independent, dependent, and what's the third one? Control. So independent is the one that you are testing. So you change this slowly as you want to. You alter this, or you vary this. So for example, if I want to see the effect of temperature on growth, I will change the temperature from 10 degrees to 15 degrees to 20 degrees to 25 degrees to 30 degrees, for example, or 35 degrees. So I can carry out the experiments at these independent temperatures as such, because I'm in control of these. Now, dependent is the one that you measure. You want to see the effect of what's happening with these temperatures. So I will measure the growth. So I might get a ruler and measure the height. I might measure the mass, it depends. Wet mass or dry mass, whatever it is. And control is where you keep the same. You keep these the same to make it a fair test. So the only thing that should be changed is temperature, right? You're going to measure the height in centimeters, and everything else, like for example the water, the nutrition, or the food nutrition, the what else could there be, the light levels, this type of soil, etc. Yeah, I'll keep all these the same, the pH levels of the soil and so on. Right, so it'll be that. Let's have a look. So what we're gonna do is I'm gonna do the CORMS. So what am I gonna do? We're trying to figure out where the birds, farmers use bird scarers. Let me read this. To make a sudden loud noise to frighten birds away. Design an investigation to find out if noise affects the amount of crops the birds eat. So what you do is the first sentence you can say, I will set up some fields of crops with noise machines. We could just call them noise machines. It's easier. And some fields without noise machines. Right. So the next thing you can write is I will use the same type of crops in the investigation. Okay, so here what you've got is you've got the bit that you're setting up and controlling. Yeah, the independent variable. The organism, you're going to make sure you use the same type of crops or seeds because they might like wheat instead of corn, and if you grow corn in some and wheat in the others, you might get more wheat eaten. Right, next one. R. How do we make it reliable? I will set up a few fields of crops with noise machines and a few fields of crops with no noise machines. Okay, so that means that that's the R done. But I always like to add this. This is just me. I will remove anomalies and average out the results. Right, once that's done, how am I going to measure the success? So what I'll do, I will weigh the crops and calculate the kilogram per kilogram per okay, square meter. I will, so what we can do is keep it the same. So I will do this. I will let the, okay, let's just reword that. I will measure the crops after 5 months of growth. Yeah, 5 months. So what you've done, you said how you're gonna do it. I will weigh the, okay, and calculate I will weigh the crops and calculate the mass per, should I put, right, let's put this in words then. Calculate the mass in kilograms per meter squared. I think that would be better. Right, so first of all, you're going to say you're going to do kilograms per meter squared, and then you're going to measure it within a time frame. If it's appropriate, it's good to put a time frame in. If you look at all the mark schemes, that's what they do. So that's our two M's. And now what we're going to keep the same. So I will ensure the same species of crop are used. Same area, same size of field if possible. You won't get exactly the same size, but it's no good having one on a square meter and the other one on an acre. Same size of field. What else is there? Yeah. And same time of year, and even in the same geographical location. So that'll do it. So that'll give you your full 6 marks. And if you're in the same geographical area, that will help because you're going to end up with the same species of birds again, because different birds might tackle different things, because some might not touch seeds, some might prefer berries. So if you've got an area where you've got loads of berries, they might not attack the seeds or the crops. So it's that CORMS there. Right, let's go. There was another one here. I saw this one. Right, I'm gonna do about 3-4 and then you can do the others, hopefully. Right, let's do that thing again. So let's put 2 M's and 2 S's. That's CORMS. Right. So now, because they've asked you to write in full sentences and paragraphs, we're going to do that rather than bullet points. Describe an investigation to find out if leaves growing at the bottom are greener than leaves growing at the top. So I will collect leaves from the what's going on? Right, okay, I will collect leaves from the top of trees and leaves of the bottom of the, let me put that up there. Hello, yeah, and from the bottom of the same trees. Right, so what we want to do is we want to make sure that I will make sure the trees are of the same species and age, because that'll have an impact. Right, I can put down here, let's just do something else here. To make sure that the test is reliable, I will pick leaves from a number of trees. I will remove any anomalies, right? Anomalies, that is strange results that are away from the normal or the average. I'll remove any anomalies and average out. Okay, that's that done. Right, so we've done your independent variable, talk about what you're going to try to make sure that you keep the same. It's normally species, how we're going to make it reliable, what we're going to measure. So what we'll do is I will measure the, okay, I will measure the, right, before we do that, what we'll do is we'll take out, let's take out the chlorophyll first. Oh, yeah, I will remove, and again now this bit you have to know from how to remove chlorophyll, and you get that from when you're testing a plant for starch, and it's one of the experiments, but I won't go into that. I will remove the chlorophyll. So remember, there's your chloroplast, that's the green bit. And when sunlight comes in, this chloroplast, this structure here, helps turn sunlight, or uses sunlight to produce glucose. Now the green part, the green pigment inside is called if I can spell it, chlorophyll. And this structure is a chloroplast. And it's the green pigment that we want. Oh, color. So I will remove the chlorophyll by heating it up, just put boiling, in ethanol. I will then compare the color. The darker the color, no, that's not how you spell color. The darker the color, our color is like this. Our American cousins spell it like this, but we don't want this. This is ours. The darker the color, the more chlorophyll there was. Chlorophyll, or darker green it was. Okay. Now what do I keep the same? I will control the number of leaves I pick. I will weigh them and use the same mass for leaves at the top and bottom. Right. Okay, yeah. I will also use the same age, and I've already set it up here, species of tree. I will also it's the time of year. I will also do the test at the same time of year. Right, that'll cover it. Right, so let me just go over that. Right, because they've said do it in sentences, and it's really a good habit. Even if you do it in bullet points, but do full sentences. And this really here is, I suppose, that's 2 bullet points. That's 3 bullet points. Sorry, this is 2 bullet points, and this is 2 bullet points. So Really, that's what you've done. You've just grouped the bullet points together. So the CORMS, your independent variable, what you gonna control? I'm gonna control the number of leaves from the top, and leaves from the bottom of the same tree, right? Let's talk about the actual organism that you're going to use. I will make sure that the leaves are of the same species and age of tree, right? How are you going to make it reliable? I will make sure that the test is reliable. I will pick leaves from a number of trees, yeah? Number of trees, because that'll make it even better. I will remove any anomalies and average out the results. Remember, we said we just add this as a one-liner every time. Anyway, then what you do is, how do we measure? I will remove the chlorophyll by boiling in ethanol. Again, this comes from that experiment that you need to know on how to measure the amount of starch, or measure the starch in a leaf. I will compare the color. The darker the color, the more chlorophyll it is. So the darker it is, the darker the leaf was, or the greener it was here. Right, and then what we're going to keep the same to make the test fair. I will control the number of leaves I pick. So it's no good picking 10 leaves from the top and 2 leaves from the bottom, and or 5 big leaves from the top and 5 small leaves from the bottom. You've got to make it fair. So what I'll do is I will weigh them and use the same mass. So I'll pick 100 grams of leaves from the top and 100 grams of leaves from the bottom, for example. Yeah. I will also make sure that the age and the species of the tree are the same, because that can impact how green leaves are. I will also make sure that it's at the same time of year. Why? Because if I measured it in autumn, there'd be less green in there. If I measured it in early spring, there would be less green in there. And in the height of summer, maximum sunlight, there'll be really dark green. Right, let's do one more. Right, let's try this one. So we're looking at human insulin. So let's have a look here. Whoa. So to produce large amounts of human insulin, the genetically modified bacteria are grown in a fermenter. Right. Describe an investigation to find out if temperature affects the amount of insulin. If temperature affects the amount of insulin made. Right. Again, they've said full sentences and paragraphs. So I put 2 M's and 2 S's if it's appropriate. Right. I will set up a number of fermenters at a range of temperatures. So for example, you might even write it down. You might say, okay, I'm going to set it up about 15, 20, 25, 30, 35 and 40 degrees Celsius, right? What you do is you make sure I will use the same species of genetically modified bacteria. Okay, so that's got them two done. And now we can add to make okay, we don't even need to write that. Let's just keep it simple. Right. I will repeat the experiment in for each, let's make it clear, for each temperature a few times. I will repeat the experiment for each temperature a few times. So what you're saying is you're going to say, remove any anomalies and average out the results. Right. Right, now what do we do? So we've done that. We're going to now look at what we're going to measure. So I will measure the insulin produced in kilograms. I will measure the mass of, we'll put it here, mass of insulin produced in kilograms in each experiment. Right. To have a fair test or investigation, where's the word? Yeah, it's there, it's normally given there. To have a fair investigation, I will I will do what? I will keep the following variables the same. So I will keep the pH levels the same. I will keep the amount of nutrition or food and oxygen levels the same. You can include other things, but that's more than enough there. So what you've got, I will keep the number of fermenters at a range of, I'll set at a range of temperatures. So you're saying you're going to set the range of temperatures. Right, is that? Let's see if we can, yeah. Right, I will use the same species, so that's the O of GM bacteria. I will repeat to make it reliable. I will then remove any anomalies and average out the results. That's that covered. How do you measure it? I will measure the mass of the insulin and just say in kilograms. I suppose what you could do is you could say you could set a time. Let's put that in actually. In each experiment over a 24-hour period. Yeah, so you keep the time the same. Okay, to have a fair investigation, so this is the control variables, what you're going to keep the same. I will keep the pH levels, the amount of food and oxygen levels the same. That's covered it. Right, I wonder if there's another one we can do. Look, they're asking for what else is there? Again, yeah, they've started asking for full sentences, so it's probably a better way of doing it that way. Design investigation, amino acids. Root growth, okay. There was one here, fungus one. There was a fungus one. No, no. Did I see a fungus one? Yeah, this is a trickier one. Let's try this one, and then we can call it quits on that. The garden bench is painted with a fungicide. Right. Fungicide is a chemical that kills fungus. Okay. This prevents wood being decomposed because the fungicide kills the fungus or fungi. There are different fungicides that can be used. Design investigation. Okay, so we want to design an investigation to see which fungicide is the best one. Full, okay, decomposition of wooden logs, right. So what we do, same thing again, right? That's to remind me. So, okay, what am I going to control? So we want to work out, so there's different fungicides. So I will use different, did you say different? I didn't say types, different types, okay, or brands of fungicide during this investigation. Right. I will. What am I gonna? Because remember, it's the wood that's rotting. So I will use the same type and age of wood. So it's no good having fresh wood and one that is old, yeah, and one that is damp with moss growing on it, because these will decay at different rates anyway. So I will use the same type and age of wood, right? Right, okay, what I would say is, let's not use the word type. Let's use the word species. 1 second, species. Because the species, the type of wood will affect it, definitely. Because some woods rot quicker than others. Right, how do we make it reliable? I will make sure I repeat each type of fungicide. I will make sure I repeat each type of fungicide on a number of logs. Just say 3 each. This is to make it fair. Remove any anomalies, and then what do we do? And average out the results. Okay, so that's that done. Right, so what we're going to measure. Right, so what I would do is I will measure the mass of wood before and after the investigation to see how much is rotted away. And just say over a period of, just say 2 years or year or whatever you want, 2 years, right? Now, finally, what would I keep the same? I can use the word control variables. The control variables I will control, yeah, are the temperature, the volume, so you keep the same volume of fungicide, not pesticide, fungicide, the same volume of fungicide, sort of oxygen levels, and just the normal moisture levels. So it's no good having it in a really dry area for one, and in a really dark and dingy area where rot's going to set in another. Right, so what we've got is, so that's that done anyway. So I'll use different types. It's talking about different types. So I would use different types of fungicide, and I will make sure I use the same species. And in this case, the age will affect it. Right, that's that done. I will then repeat using 3 logs for each, and I will again remove any anomalies and average out the results. That's a standard. So that is a standard, yeah. What will I measure? The mass of wood before and after the investigation to see how much is rotted away over a period of, and because rot takes a bit longer than normal, you leave it, you won't say it, oh, 2 hours or 2 months or 24 hours, right? And what am I going to control to make sure it's fair? Like things like temperature, moisture levels, oxygen levels for aerobic respiration affect things, and it's no good putting a little bit of fungicide on one and loads on the other and saying, oh, look, guess what? It works really well on brand B. That won't be fair. So keep the volume of fungicide the same. So you've got enough there for your 6 marks. Right, another one that you could try out is this garlic one. Right, I'm going to call it quits there. You can try the rest now.